Yep.
Welcome. It's a great pleasure today to present Erin Gorsich to you for her doctoral dissertation seminar. Erin came to OSU in 2008 when I was recruiting a couple of students for the then newly funded project on buffalo and their co-infections, which is also what Erin went ahead to study. Erin grew up in Ohio, and for her undergrad, she stayed close to home and went to Denison College. And Erin came really rather highly recommended from Denison. She studied biology there and a minor in chemistry and also quite a lot of math, and came with a magna cum laude degree from Denison. But that's sort of normal, right? Well, Erin had also done all this charitable volunteer work. And she was a great athlete. She was tennis captain at her college and athlete of the year, and they gave her a president's medal. It was really quite intimidating, and it had intellectual merit and broader impacts written all over it. It later did turn out that the NSF just loves Erin, so I was right about that. So anyway, back in 2008, I think it must have been January, I invited, it could have been the December before, I don't remember, and it was winter anyway. I was very happy to invite Erin for an interview here at OSU, and she arrived, and what was really quite striking was that the intimidation, I guess, went both ways. And despite the fact that we were experiencing a normal Oregon winter, not like this, Erin really did want to come here, but she didn't really want to talk to me. Not until about 2 or 3 years later, when she got over the shy. So I know I'm sounding like an old aunt now, but she really was young and shy. But it wasn't too hard to see past that, to the bright little flame of curiosity and intellect and scientific passion that was there, and that just needed a bit of fresh air to fan it into a bonfire. Luckily here in Oregon we do have a lot of fresh air, and in South Africa, Kruger Park, where Erin went for her fieldwork, there's even more fresh air. And so I'm proud to say that Erin definitely did have bonfires, scientifically speaking. So throughout her PhD then, Erin spent some time here in Oregon, but she also spent more than a year in Kruger National Park in South Africa chasing buffalo for her fieldwork and doing most of her associated lab work in our field lab facilities there too. She also went to the Netherlands for a substantial amount of time to work with our theory collaborator there. And so in sum, she's ended up with a really nicely rounded dissertation that has theoretical aspects, fieldwork, lab work, and some really rather hairy statistical analyses. I'll give you a mea culpa right here. I thought it was a good idea to present Erin right when she got here with a nice data set from a previous buffalo study, so that she'd have something to get her teeth into before she had her own data, not realizing how complex that data set really was, but it formed the first chapter of Erin's thesis in the end, and she did a really nice job. So with that, I'll give the stage to Erin.
Collaborators. So parasites are arguably one of the richest trophic groups on the planet, and their diversity encompasses all major taxonomic groups, ranging from scores of tiny viral and bacterial infections presented on the top here, to less tiny but equally elegant fungal and protozoan parasites, including this Giardia parasite represented here, to the less charismatic and definitely less elegant representations from the arthropod and worm taxa here. Their ubiquity is so pronounced that in natural populations, infection with multiple parasite species or co-infection is the norm. A recent review estimated that the average mammalian host species was infected with up to 8 extracellular parasite species at once, and that's just the ones on the bottom half of the screen. So once you start adding on to that complexity, the overwhelming number of viral and bacterial infections that we know to be simultaneously infecting one host, pretty soon my cute charismatic megafauna on this photo looks less like a buffalo and more like a diverse community of parasites And one could argue that that's a community of parasites that should be subject to some of the same rules and approaches that we would apply to other ecological communities. So for example, parasites within this community could interact to increase or decrease each other's spread. One example of this came from an epidemiological study in Kisumu, Kenya, represented here, where they found nearly 1 million excess malarial cases due to co-infection with HIV. That's 10% of the total malarial cases that they found in this town. So increased malarial prevalence due to HIV is likely caused by the well-known immunosuppressive effects of HIV on the host. And as a result of such observations, the World Health Organization has changed their policy, or has suggested policy, to focus bed nets and prophylactic drugs to those patients that are infected with HIV. And the pictures shown here are from the cover photos for the HIV and malaria co-infection programs from the Bill and Melinda Gates Foundation and the CDC. But co-infection may also alter the severity associated with an infection, thereby alter the disease progression. So one iconic example of this is TB nematode co-infections, where when a patient is simultaneously infected with intestinal parasites, they have altered immunological responses to TB, increased severity of TB, and it even interferes with the treatment of TB. And so therefore it's pretty clear that co-infections can have pretty significant effects for human health. And mounting evidence suggests that this may be important in wildlife populations also. So why then study wildlife, or study co-infections in wildlife? So even though a lot of the background information by its very nature comes from human medicine, as an ecologist, I'm interested in things like how parasites regulate populations, how environmental factors affect the transmission and persistence of pathogens in the wild, and how host movement and behaviors affect that persistence of the pathogen. And so this work uses African buffalo as a model system to ask disease ecology questions, because for studying diseases, they're a very practical system. And they're a very practical system for the same reason that they're important. Buffalo often get and share a lot of pathogens with cattle. And as a result, a lot of the immunological methods and diseases and background information from that system can be easily applied to our system. They're also a good study system because they're very parasite rich and ecologically important. African buffalo live in large herds, anywhere from 100 to 600 individuals. And because they're so gregarious, they're often infected with a bunch of parasites. They live in a whole range of savanna habitats, from completely open to very dense thickets, to somewhere in between, like our beautiful field site picture here. They're bulk feeders, which allow them to live in such a range of habitats. And because of this behavior, it makes them very essential components of savanna ecosystems. They allow the persistence of other herbivores and really create different heterogeneities in that habitat. And so I study buffalo not just because I care about human infections, but because they're a really tractable system to ask disease ecology questions. They're always infected with something really interesting, and their behavior and ecology are really well known, so you can apply disease ecology questions very well to this system. So I use African buffalo to look at the interactions between two pairs of interacting pathogens and parasites. The first pair is interactions between nematode parasites, so your gutworms, and Coccidia parasites, another intestinal parasite. In the interactions between two bacterial pathogens, Brucella abortus, which is above, the causative agent of brucellosis, and Mycobacterium bolus, the causative agent of bovine tuberculosis. I'm going to be referring to bovine tuberculosis as TB for this talk. A little different than human TB, but still TB. And so I use these two pairs of parasites and pathogens to really ask how and when does co-infection affect host fitness, host populations, and the dynamics of subsequent pathogens. So in my first chapter, I examined the host fitness costs of infection and co-infection with intestinal nematodes nematodes, and intestinal toxicity of parasites. And I examined these fitness costs across the wet and the dry season. In my second chapter, I got to jump into the TB brucellosis system, so I had to start at square one. So in my second chapter, I evaluated a diagnostic test so I could be able to distinguish between infected and uninfected buffalo with brucellosis. In my third chapter, I applied the tools from chapter 2 to understand the host fitness and host population consequences of infection with brucellosis. And in my final chapter, I asked how tuberculosis altered the dynamics of brucellosis infection, and I tested whether that happened, whether that modified the host fitness effects or the host immunological effects. And so for the purposes of the talk today, I'm going to be focusing mostly on chapter 2 and 3, but if anyone is interested in intestinal parasites or diagnostic methods, I'm more than happy to talk about them. So in these chapters, I use tools from disease modeling to combine the individual level effects of brucellosis that I identified in chapter 3 with the cost of co-infection that I identified in chapter 4 to really ask how co-infection alters the dynamics of a subsequent pathogen. This study was conducted in Kruger National Park. Kruger is located in the northeastern corner of South Africa, right on the border of Mozambique and Zimbabwe. And in this system, tuberculosis is an emerging disease. It was introduced on the southern boundary of the park in around the 1960s. And since then, it's been increasing in both prevalence as well as spreading in range, so it's spreading northward throughout the park. And as a disease that can infect livestock and human populations in this area, especially because it's a high HIV-infected area, its maintenance in buffalo populations is considered problematic. As a result, the fitness effects of TB are fairly well characterized. TB in buffalo is primarily a disease of the lungs, just like human infection. Human infection with TB is, and it results in declines in condition and reduced survival in old and young buffalo. This picture here is a healthy lung versus the normal lung. You can see that the healthy lung is smooth and light. But what happens throughout the course of TB infection is your immune system walls off the infection in these structures called granulomas. And as a result, a lot of the tissue becomes unfunctional. And if you actually lift up a buffalo lung that's been infected with TB, it becomes really hard and really heavy. So although the costs of tuberculosis infection are quite gross and high, it's pretty well known, the costs in buffalo. What's not known is how TB infection will affect other native pathogens in the buffalo pathogen community. One such pathogen whose transmission and fitness effects are likely to be altered by tuberculosis is brucellosis infection. I thought TB's influence on brucellosis would be particularly interesting because they're modified by similar immunological mechanisms. However, unlike TB, the fitness effects for brucellosis are not well characterized. We know from cattle and bison that brucellosis is expected to cause abortions a couple years, for the first few years after infection, and declines in condition. People have also noted these arthritic swellings called hygromas. But it's really not thought of that big of an effect. I don't know, that looks kind of serious to me, and an abortion seems kind of painful. So perhaps something more is going on in the system. And of course, we didn't really know the rates at which these things were happening. I didn't know how frequently the disease was causing decline in fecundity, or how much the condition costs were. And so before I could really ask how TB was modifying brucellosis, I first had to ask what was going on with brucellosis. So I can represent what is known and unknown in the system mathematically with the disease dynamics model. This figure shows the framework of a disease model. Here, S represents the number of susceptible animals in the population. I represents the number of infected animals in the population. And those animals are the ones that are having abortions and shedding the bacteria into the environment, so they're infected and infectious. Latent represents those animals that are infected with the pathogen but aren't shedding the bacteria. So don't get too caught up on infected versus latent. both are detected as positive, but this just allows me to explicitly represent what's going on with transmission. So this representation allows me to explicitly represent the fact that animals become infected with the pathogen at a known rate, and we call it the transmission rate beta. And after animals are infected for a few years and are having abortions and shedding the pathogen, they become latently infected with, again, a known rate. What I don't know about this system is the fecundity effects of brucellosis. So I don't know how infection with this pathogen affects the birth rate into the population that's represented by B. And I don't know the survival effects of brucellosis. So the mortality rate, which I represent as mu. So I don't know the difference between the mortality rate and susceptible and infected. So for the talk today, I'm going to walk through the results and methods that I use to estimate these survival and fecundity effects of brucellosis, and then talk about how I determine whether TB would modify the system. I was able to ask these questions by combining two really extensive and wonderful data sets that I had the opportunity to work with. The first was a park-wide disease surveillance data set that was collected by veterinarians at the State Veterinary Services of the park. And so the park veterinarians collect routine information on all the major diseases within the park. So they have baseline information to look at how diseases are changing over time and what the spillover risk is with those diseases. So this is a really geographically extensive data set, but not very detailed. And so I combined this data set with a long-term buffalo monitoring project that was established by Dr. Jolis and collaborators. And this data set monitored 100 buffalo in two interacting areas within the southern portion of the park. And so these buffalo were monitored every 6 months, so we would have detailed information on infection and immunity. A day in the collection of this data set looks a little bit like this. We would wake up unnecessarily early so we could be at the buffalo by dawn. We would find the buffalo by Joe Spawn, would track them, and we would track until either we found the buffalo or till her arms got tired, which took a really long time, some days. As you drive up to a buffalo herd, you can see like this that most buffalo are undisturbed by the project. If you got really good, you could detect the buffalo quite easily. You can see that the buffalo we're monitoring are wearing radio collars so we could find them, as well as brands on the bum. Those buffalo were chemically immobilized, and once they were immobilized, we collected a range of demographic and infection data. We collected fecal samples for intestinal parasite burdens. We collected blood samples to get serological infection status and immune data. We assessed host age by tooth eruption patterns and wears of their teeth, and host body condition by palpation of various areas where the buffalo store fat on their ribs and their hips and their spine. Once we were finished, immobilization was reversed, and our buffalo went back to her herd until the next 6-month time step. And we would take the blood back to the lab where it was processed by various collaborators. So I combined these two rather extensive data sets to estimate the spatiotemporal patterns of brucellosis infection and the fitness effects of brucellosis infection. And these two bits tell me the proportion of animals that are susceptible and infected, as well as the rates associated with the new parameter circle K. So first, to determine the spatial and temporal patterns of prevalence, I compiled 6 years of the park's surveillance data. And this figure shows the sampling locations that occurred in this time period. So each red dot on this map represents a herd that was sampled, and it resulted in about over 1,600 sample points. I used generalized linear models to examine the age, sex, spatial, and temporal patterns of infection. And surprisingly, I found absolutely no temporal pattern over time. Nothing changed over time. But I did find a rather striking geographic pattern where prevalence was very low in the northern part of the park at about 14%, but was high in the southern part of the park at 26%. And this pattern was largely driven by underlying geological variation related to resource availability. Infection also showed an age prevalence pattern typical of a chronic disease. When you plot the brucellosis prevalence by age on the x-axis, you'll see a hump-shaped distribution. In this graph, the light bars represent females, and the dark bars represent prevalence in males. The letters indicate significant differences among age groups for females, and the stars indicate significant differences between males and females within a single age group. So in females, infection increases until adulthood, and then it kind of decreases over time. This is typical of a chronic disease and often indicates mortality in older animals, or perhaps a reduction in our ability to detect the disease at that time. We also found a really sharp contrast in sexes and prevalence in the sexes at calfhood, with high prevalence in young calf males. Although we don't really know what's going on here, we have a couple good guesses. We think that this is related to maternal immunity, and this is immunity that the mother provides to her calf for the first 5 or 6 months after birth before they can develop their own immune response to keep them protected. And so this could perhaps be due to the duration of maternal immunity and the time that we capture the buffalo. So if mothers are giving birth to males later in the season, and we are doing the captures also late in the season, we may still be detecting maternal antibodies in males, but not in females. This pattern could just also be due to the fact that mothers differentially partition those maternal antibodies differently to males and females. So next, I asked, well, what are the mortality costs associated with brucellosis infection? Are there any mortality consequences? And to do this, I use a cost proportional hazards analysis. This should look familiar to anyone who has seen a presentation from the Blaustein lab. This analysis estimates the proportional difference in mortality among treatment groups, any potential predictor variable that you're interested in. And so I use this analysis to ask whether there was a difference in mortality among susceptible and infected animals, so a difference in that mortality rate mu on my diagram. These results plot the proportion of buffalo surviving on the Y by time on the X, and you can see that the curve representing brucella negative buffalo, the dark line, has increased survival or reduced mortality compared to infected animals. So brucellosis is associated with a higher mortality rate, and we estimated that mortality rate to be about 3-fold higher than uninfected buffalo. We also saw a drastic increase in mortality in one of our herds compared to the other herds, about 4-fold higher in the herd called the crocodile bridge herd than the other herd. And in that herd with high mortality rates, we saw really little effect of brucellosis. So environmental variation here is really important. So then we asked if there are fecundity costs associated with brucellosis infection. So we're really looking at whether there's a difference in that birth rate associated with infected and uninfected animals. And to do this, we use generalized linear mixed models. And this statistical technique directly avoids problems with pseudo-replication associated with our longitudinal data set. So we account for the fact that we're catching animals multiple times by representing animal ID explicitly in our models. So anytime I'm not talking about survival analysis in this data set, I'll be using mixed models and accounting for animal ID. What I found was that after accounting for herd and age and season, brucellosis caused reduction in the likelihood that we would observe in calves. So if animals were infected with brucellosis, they were less likely to successfully calve. We also found that in the herd where we saw the highest mortality rates, that there were the lowest fecundity rates. And we also saw this strange condition by brucellosis interaction term that becomes clear if we plot the probability of that buffalo being with a calf by body condition. The negative slope on this calf reflects the fact that having a calf and lactation and pregnancy are really costly activities for the buffalo. So throughout the course of the pregnancy, animals typically lose condition over time. So there's very few animals that are in high condition throughout their pregnancies. Most buffalo, however, reduce condition and are in a very low body condition, and it's in those animals with low condition where the effect of brucellosis is highest. So if you are doing really well and can manage to stay in good shape throughout your pregnancy, then the effect of brucellosis is almost negligible. But that's really rare. So I then combined those individual level survival effects and fecundity effects of brucellosis to population level patterns using a Leslie population model. So briefly, a Leslie population model summarizes age-specific survival and fecundity rates into one big matrix. So don't be scared. All that this is representing the fecundity rates for each age on the top rows of this matrix and the survival rates on the bottom. And this summary represents the increase in population growth between an initial time point and a subsequent time point. So our data directly estimated those rates. And I had specific survival and fecundity rates for each herd for multiple conditions and with and without brucellosis. So all I had to do was plug them in and see what happened to population growth. And so in this figure, population growth is represented with lambda. You can interpret lambda as if it's greater than 1, population growth is increasing, whereas if it's less than 1, population growth is decreasing. So by comparing the effect of brucellosis across multiple herds, multiple conditions, based on the statistics that I found in my statistical analysis, I came to 3 really rather exciting conclusions. The first one is that environmental variation is important. The population growth rates are drastically different among herds. So in the crocodile ridge herd on your left, population growth is always decreasing regardless of what's going on with disease. And in the lower side herd, it's always increasing regardless of what's going on with disease. So very different things are happening in these 2 herds. The second conclusion is that population growth does actually decrease with increasing disease prevalence. So all of these lines have a negative slope. As brucellosis increases in prevalence, more animals are suffering from fecundity and survival costs of infection, and you get an overall reduction in population growth. And finally, and a little bit counterintuitively, the slope of this line in the lower Sabi herd is much stronger than in the crocodile ridge herd. And so the effect of brucellosis on this population is much higher than in the crocodile ridge population, even though it's in overall better shape. So perhaps the effect of a chronic disease might be more important in a herd that's doing kind of okay than a herd that's doing really, really bad. And so we kind of think that in this herd, the really high mortality rate that we saw means that they're dying from so many other different things that they're not waiting around to suffer from a chronic disease. Okay. So in summary, we found that prevalence varied spatially throughout the park. So we now have an estimate of the relationship between the number of susceptible and infected animals, and a range that's for that. We found that brucellosis was associated with a higher mortality rate in infected than uninfected buffalo, although this varied by herd. And so we now know that this mortality rate in infected animals is higher than in uninfected animals, and critically we know the magnitude of that difference. also know that brucellosis was associated with lower CAG success, so we know something about the birth rate into our populations now. And finally, when you combine these two rates together, you can have pretty significant consequences at the population level. So now, to determine the effect of TB on brucellosis, we estimated its effect on a few key parameters. Those are the transmission rate, we call it represented by beta, so we're really interested in how fast animals are becoming infected, and the mortality rate, so whether TB results in increased mortality or minimal mortality. Okay. So tuberculosis infection is associated with reduced condition. So perhaps if animals become infected, lose condition, they may be associated with increased mortality. Sorry, I did this backwards. So perhaps they lose condition, and so they're more likely to become infected with a subsequent pathogen. So you lose condition, you're feeling really bad, you get exposed to another pathogen, you're more likely to get infected with it. So the rate at which animals are moving this way on the schematic diagram is increased, and so we expect to see an overall increase in prevalence because more animals are infected. But these two pathogens are related by really similar mechanisms, so perhaps immune activation, especially during the early stages of infection, could allow you to have decreased transmission because those effector mechanisms might be beneficial to both pathogens, and in which case we expect to see reduced movement into the left side of the schematic and overall decreased prevalence. But as I hinted at earlier, tuberculosis may also affect the mortality rate in those animals infected with brucellosis. Again, if TB causes really strong declines in condition, you can see increased mortality in co-infected animals, and you're losing those animals infected with brucellosis, and so we expect the overall brucellosis prevalence to go down. And the reverse, if the immune response that are beneficial to both result in decreased mortality, we might see an overall increase in prevalence. And so in the absence of really strong a priori hypotheses, we really wanted to know what the net effect of both of these two parameters were on prevalence. And so again, I was looking at how TB invasion in the system might modify the dynamics of brucellosis infection. And I did this by estimating the effect of TB on transmission, that's the beta, survival or mortality, that's the mu. And I did this and then I'm going to combine these two rates into a disease dynamics model. So first, is TB associated with increased susceptibility? To ask this question, I again use the cross-proportional hazards analysis. This is just like the mortality analysis, except for the event that we're looking for now is infection. And I did this analysis in two stages. First, by considering all potential variables, and then by kind of honing in on those animals that had really detailed information on TB, the course of TB infection, and asking whether those susceptibility rates changed throughout the course of an infection. So I then compared tuberculosis as a binomial variable. Are you infected with TB? Are you not infected with TB? As opposed to a categorical variable. Are you infected with TB recently? Or are you chronically infected with TB? Have you been infected for a while? Or are you just not infected at all? What I found was animals that were infected with tuberculosis had a 2.8-fold higher rate at which they became infected with brucellosis. So TB is increasing these animals' susceptibility to a subsequent pathogen, but the magnitude of that effect is really limited within 1 year after infection. So only in acutely infected animals are you seeing an effect of brucellosis. Okay, so now that's the transmission parameter. So now we're going to move on to the mortality rates. And I asked, is TB associated with altered mortality? Does it increase or decrease it? Again, I did the same cost proportional hazards analysis, and again, used that two-stage approach to compare whether the effects of TB on mortality were consistent throughout the course of infection. What I found was the effects of both pathogens were consistent throughout their infections, and both were associated with increased mortality. So this figure shows the survival again on the Y and time on the X. And you can see that the dark line again represents those animals that are susceptible to both pathogens. And both pathogens are additively associated with increased mortality. So when you're co-infected, your mortality rate is way higher. So in summary, both TB and brucellosis are additively associated with increased mortality. Okay, so we found that tuberculosis increases transmission, and it increases transmission quite seriously, but only for a year after infection. And that tuberculosis increases the mortality in those co-infected animals. And then we scaled up these individual level effects to the population level by using a disease dynamics model. And we did this extra step of analysis because it's at that population level that managers are doing their disease surveillance. So here's the working model. It looks a bit like this, where I explicitly represent those animals that are infected and co-infected. And don't be scared. I'm gonna brazenly skim over all this map with the caveat that you please talk to me about it later if you're interested in it. I usually sit in my office and would love to have someone to talk about this. So here's what I'm gonna walk you through the specific assumptions that are reflected in this map. So in this representation, I represent those animals infected with tuberculosis here, where S is still the susceptible animals. Animals can either be acutely infected with TB or chronically infected with TB, and I really had to make it this way because in our susceptibility analysis, we found that the effect varied over the course of TB infection. Here's the schematic diagram that I've been showing you the whole time. This is brucellosis infection group and over population. So I'm just adding layers of complexity here. And I represent the co-infected animals with I-co and L-co here. So this representation allowed me to represent the fact that the susceptibility to brucellosis infection is increased in those animals with TB. So I let this rate be higher than the rate at which animals become infected with brucellosis without TB, and that higher than the rate that animals become infected with tuberculosis or with brucellosis, when they're chronically infected with TB. This structure also allowed me to represent the duration of that increased susceptibility to last that it lasts 1 year, just like our statistical analysis showed. And finally, our stats told us that the mortality rate in co-infected animals was additively increased than in singly infected animals. And so I let the mortality rates in those co-infected buffalo here be higher than the mortality rates in the singly infected buffalo here. And those mortality rates again be higher than those in the susceptible animals. That's scary. And so this model allowed me to make two really exciting predictions. The first prediction is that brucellosis should be more common in animals co-infected with TB. And so this figure represents brucellosis prevalence on the y-axis and what happens as you increase susceptibility to brucellosis with TB infection. So in the absence of TB, the equilibrium brucellosis prevalence was 30%, and I said this. And so given the mortality rates that we observed, if there's no increase in susceptibility also associated with infection, prevalence drastically decreases. And this should make sense. All the animals that are co-infected with TB are dying, so you see a really low prevalence here. And it's simply because we're representing mortality but not transmission yet. And so now I'm going to show you what happens when you represent the susceptibility in the parameter space that our statistical models told us. So the confidence interval around, or the estimate for increased rate of new infections was 2.8, with a confidence interval from like 2 to 4-ish. So that's what's represented here. And as you can see, prevalence drastically increases back up to about what it was before. And critically, that those animals infected with tuberculosis now have a much higher prevalence than those that don't. And this playing is very nice, but I wanted to know whether this was actually reflected in my data. And so I compared the probability of being infected with brucellosis with and without TB. And what I found was that, well, yeah, animals with TB had a much higher prevalence than those without TB. In fact, of the 549 buffalo sample points that were TB negative, brucellosis prevalence was 30.7%. And compare that to the 221 sample points that were TB positive, prevalence was 52.9%, much higher. And remember, I did my statistics after. So these represent buffalo sample points, but I did my statistics accounting for animal ID and a bunch of other cofactors. So this result is really robust. In fact, after you account for age, it's still quite significant. And so you can see that in the light bars of those animals infected with TB, brucellosis prevalence is much higher than those co-infected buffalo, regardless of age. So in summary, in our data, we also showed increased prevalence in co-infected buffalo. The second major result from the model is that if you're just looking at the population level, after tuberculosis invades, you may see no effect on brucellosis prevalence. This figure shows brucellosis prevalence before and after TB invasion. You can see that there's almost no pattern, no difference in these two prevalences at all. And this is really important because if you are a manager looking for the effects of an invasive disease, and you're worried about the stability of your populations, and you're just looking at prevalence patterns, you're going to miss these really drastic changes in mortality and transmission that you're seeing. So at the individual level, what's going on is very different than what you may see at the population level. So in conclusion, I asked some specific questions about brucellosis, and then I asked how TB may modify these patterns. So for brucellosis, I asked, are there spatial or temporal patterns of brucellosis infection? We found no temporal patterns, but we found a really strong spatial pattern with prevalence from 14 to 26%. I also asked, does brucellosis affect host fecundity, that birth rate there, B? And found, yeah, it has pretty drastic effects, and the magnitude of those effects are much higher if you're in bad shape overall. And finally, I asked, does brucellosis have associations with changes in mortality? Very clearly. And those effects vary drastically among herds. And when I combine these effects on fecundity with the effects on survival, I found that regardless of these context dependencies, the disease can still play an important role at the population level. So then I asked, what is going to happen when tuberculosis invades the system? So I asked whether TB is associated with increased susceptibility, that beta parameter here. Yeah, and the magnitude of the increase in susceptibility associated with TB was really concentrated on that the first year after infection. I also wanted to know whether TB was associated with changes in mortality patterns. And we found that this effect of tuberculosis was to increase mortality, and it did so consistently throughout the course of infection. And finally, I whether TB infection alters the dynamics of brucellosis infection, and the answer to this question was a little bit more complicated. So I found that at the individual level, tuberculosis does drastically alter what's going on with brucellosis infection. Clearly it increases susceptibility, but it also increases mortality. So I represented these transmission and mortality estimates for my statistical models in a disease dynamics model. What I found was some really confusing results. I found that nothing was going on, except for maybe a positive association between TB and brucellosis. They seemed a little contradictory, but it turns out that's exactly what's going on. I found no significant in my surveillance data that I borrowed from the veterinary services at the park. I found no changes in prevalence over time, even though TB has been increasing in the system. And I found an excess of co-infected buffalo, lending some support to my theoretical model. So when transmission and mortality counteract each other, disease surveillance data may miss some really important and drastic changes that are going on at the individual level. And I think that this really important result emphasizes the importance of using longitudinal data, so data that's collected over time for disease surveillance. And I also think it emphasizes the importance of considering the other parasites within the parasite community if you're concerned about the effect of the disease on a population. And so my PhD work looked at the interactions in the top right corner of this figure. But really that's just the tip of the iceberg that's going to come out of this buffalo project. There's a whole complex community of parasites that's still to be analyzed and that you can look forward to hearing about. I think Brian Henricks is taking a more community approach to the system and looking at major perturbations in the system and at the whole community. Joe Spahn in the back is looking at some host physiology and how that affects this community. And Rob's using these parasites as an excuse to look at buffalo movement and behavior and management in the system. So there's going to be a lot of cool talks coming out of the rest of the data on this project. And so with that, I'd like to thank all kinds of people. I was very well funded for this project, and it allowed me to live in South Africa and live in the Netherlands, and taught me a lot of wonderful things. But I'm also really thankful to the Environmental Science Department because their funding allowed me to travel and go to conferences before I got any of this other funding. I really appreciated that. I also have a lot of co-authors to thank. Roy Bingus in the top corner of the fishing rod and Paul Cross are responsible for my surveillance data that I use. Vanessa Azenwa, also in the top corner, is the leading parasitologist on the project. I didn't know anything about intestinal parasites before I started this. Rampal Etienne, the guy also in the top corner, is who taught me how to use models in general and how not to be afraid of them. And Bree Beechler over there, who you probably all know, has helped collect most of this immune data that I didn't get to show you today, but a big role in my PhD. There's also a bunch of South African National Parks research staff and veterinary staff that did all the buffalo captures. Rob and Jill, graduate students here, collected all the data for this project. They ran and managed the buffalo collection project. And I also wanted to thank Jane, who, well, so as an environmental science student, I get the benefit of not really having a home department. And so I can use the biomedical science department and the zoology department, the good parts of both departments. And so it also means that I didn't know what was going on in either department. So I had a lot of help. Sarah, Tracy, and Tori in zoology, and Jane in biomedical sciences. They're both wonderful. Okay. fatty part. And so I was trying to think of a good way to express how great of an advisor Anna is. And I'm not sure if I completely did it justice. But so thank you, Anna, for being such an encouraging and wonderful advisor. So I thought that I would, instead of putting lots of work pictures on the slide of Anna, remember all the places that she's given me life and science advice. So if you're a member of the Dualist Lab, you know that you're probably going to have to talk science in strange places. So Anna has the ability to teach you about life while doing field work. While hiking, you better be able to, once we interviewed a postdoc, I took him on a 7-mile walk. That was the first time I had ever gone snow showing. I don't know if you know that, but I'm going to let me. While drinking beer, Anna thinks that we should be able to talk science in a subtropical location, probably overlooking a river. Something to look forward to. And at mile 18 in a race, all are very memorable, and I appreciate them all. I'd also like to thank the members of the Dualist Lab, again, who you have to be able to talk science to in really crazy locations. I really do think they have a very special lab, and I appreciate you all very much. Heather, you and your cat.
Seamus made his contribution.
And also thank you to the zoology department who is out in court today. And I remember talking to Allison the other day about not being a zoology graduate student, but feeling very much like one. And I very much appreciate that. Yeah, it's good for mental sanity. Thanks, Brian and Angela, for getting me through my first term of anatomy and physiology this year. And thanks Allison and Sarah and Lee for all your thesis and great help this term. And everybody else who contributed to my mental sanity over the course of my PhD. Thanks, Paul. I'm sure we're talking about super spreaders there. And thanks, Mom and Dad, who flew all the way from the Midwest to come see me today. And have been wonderfully supportive. And I have lots of other sentences to say, but I'm not going to be able to hold it together. So thank you.
Questions?
So how do you know that it's the effect of TB on brucellosis and not the other way around? And like, does it matter which the buffalo are infected with first? Like, is there a sort of a history to the effect of TB?
I'm sure there's an impact of history. And when I did this for real, I looked at the effects of both pathogens on each other. So I have a paired analysis of TB on brucellosis and brucellosis on TB. And I also have, and I think your question emphasizes the importance of the immune mechanism that I didn't even get to talk about today. So we also have really detailed immune data. And so I know that the effect of TB on immunological parameters important to susceptibility and progression are much stronger than the effect of brucellosis. So that's why I decided to present it this way instead of the other way. And in fact, like statistically, that's what we see also.
I have a question about the prevalence that you found different prevalence in brucellosis spatially. And you said it could be about the environmental conditions where they were poor nutrition perhaps in one area or something like that. I was wondering if you could clarify whether you thought, whether the prevalence was lower where, or higher where the nutritional status might have been different, or if you could clarify the pattern.
Yeah, and I was intentionally vague because it's a complicated pattern. The park is kind of divided into really, okay, the park is quite complex, but people tend to divide it into two main soil types. One that is a little bit more nutrient poor, and that's a little more nutrient rich-ish. It's a bad simplification, but on the side that's nutrient poor, we see much more variation in brucellosis prevalence by other factors. And so in those nutrient poor areas, we see lots higher prevalence. Yeah, and I don't have a mechanism for that. I just have those cross-sectional patterns, and that's the best I can do right now. I would love to go back and do something like fidget stuff with it, figure out what's going on, how nutrition is driving the changes in it. Like, we've got immune data, so you could make that link between nutrition to immunity to disease, but I just haven't gotten there yet.
So you talked about that there were some environmental factors that played into the difference in lambda between herds. Did you look at herd size at all, and did that have an effect, and the rate of lambda change with the nice prevalence?
I didn't look at herd size because herd size is complicated. It varies over season, and we really have got like two distinct chunks. And I think the person who talked to me to answer that question is going to be Rob whenever he gets around to getting to that data, because he has rough estimates of herd size. But I don't really think I think that it's too confounded right now, because herd size largely reflects those environmental characteristics. So you see that spatial pattern that I was talking with Julia influences buffalo density in those areas, which influences buffalo herd size in those areas. And so one of my questions, I was trying to tease apart these things, and it's just too confounded for the 6 years of data that I had. But I would love to collect 10 more years of data so we can tease those apart. But
I think if I understood correctly, you talked about maternal immunity. You mentioned it could be different for males and females. And I was wondering if you could expand on that more. Are their behaviors different when they're young? That the maternal immunity would be, you hypothesize, could be different for males and females.
yeah, and the theory for that comes from some evolutionary biology stuff where people have shown that in some species, well, in buffalo, I think, in ungulates especially, when resources are really bad, animals tend to invest in females because they're kind of a sure bet, like they're always going to reproduce sometimes, and so it's a good fitness investment. Whereas when things are really good, like when you're in really good condition as a mom, if you invest in a really good male, then that's going to be good. And so it's all theory about why we predict, we could say that perhaps they're investing in males for females.
Okay, well, if there aren't more questions, before you all run off, I would just like to point out that Erin's party is going to be tomorrow evening, not this evening. Because she still has to do an A&P exam in the morning, so her level of relaxation isn't going to be appropriate to a friend's party tonight, but it will be tomorrow night. And so that party will be at my house, and I'll write the address up here. If you get lost, you can